Hi, this is Dr. Lauren Launen, and this is a short lecture addressing Mendelian genetics and specifically focusing in on dihybrid or two-gene crosses. Again, we're sticking to simple genetic systems where we're tracking genes that are on different chromosomes, where the genes are in two possible allelic variants. One is fully dominant to the other. Mendel worked with pea plants, and Mendel studied 7 different characteristics. So it was, he was able to pick 2 of those 7 and study their inheritance simultaneously. So in this example, he is looking at plants, at pea plants, and he's focusing in on seeds, the pea seeds, and he's looking at both their shape and their color. So the shape can be either round, which is associated with the dominant big R allele, or wrinkly, kind of lumpy looking, associated with the little r recessive allele. The seeds can also be either yellow, associated with the big Y dominant allele, or green, associated with the little y recessive allele. So he's tracking the inheritance of both those traits, and therefore the genes controlling them at the same time. It's important to remember that the R and the Y gene are on different chromosomes in this particular example. When you have 2 parental type organisms of the genotypes shown, they are each able to produce only the gametes shown because they are homozygous recessive for both, or homozygous for the genes that we're tracking here. So this big R, big Y parent can only produce big R, big Y containing gametes. That's the genotype of the gametes. This little r, little y parent can only produce little r, little y gametes. That's the genotype of the gametes shown. When these fuse during fertilization events to give you the F1 individuals, the F1 individuals will all have uniform genotype. They're the double heterozygotes shown here, and genotype. They'll be dominant for the traits that are being tracked. Where it gets more interesting is when this F1 goes on to reproduce. This F1 double heterozygote organism looks like this: big R, big Y, or at least that's its genotype. It looks like it's yellow and it's round. That's its phenotype. That organism can produce all 4 types of gametes shown, and it will produce them in equal abundance or at equal proportions. So 25% of the gametes in that F1 generation will be this, another 25% this, another 25% this, and another 25% this, in equal abundance if R and Y are on different chromosomes. Those gametes can be laid across the axes of a Punnett square in the manner shown in this image, and then what you fill in the Punnett square is the F2 or second filial generation. When you fill in the Punnett square, you're mimicking a fertilization event. You get genotypes from that. With your knowledge of the genetic system, you can go back, look at each genotype, and assign a phenotype. Wherever you see a big R, either in 2 forms as here or in 1 form as here, you know that you're going to have round as the phenotype. Wherever you see a big Y, either here in 2 big Ys, or down here, 1 big Y, because it's Y, big Y is fully dominant to little y, you know that you're going to have yellow seeds. Down here, we've got little y, little y. So we're going to see the recessive form of both traits being tracked, wrinkly and green. And so you can go through and you can fill in all of the genotypes and all of the phenotypes. Most common student error in this circumstance is not correctly understanding that each gamete should contain only one of the genes being tracked. That's what it means to say that we have haploid gametes. So 1 R, 1 Y. Therefore, anytime you're looking at the Punnett square, it's depicting diploids that result from fertilization events. You should then get back to having 2 R's and 2 Y's in some form. So you should never see like RR, YY, for example, when you're looking across the axes of the Punnett square. Back to the patterns that we expect in this F2, we would expect to see 9/16 fully dominant, 1/16 fully recessive, 3/16 and 3/16 are the combination of dominant and recessive, the two possible combinations shown here. And that is a characteristic of a Mendelian inheritance pattern when you're looking at two Mendelian traits that are inherited according to the patterns that Gregor Mendel discovered back in the 1800s. Mendel also did test crosses. So if his hypothesis was that the F1 had this double heterozygous genotype, he could test that hypothesis by looking for the 9:3 to 3:1 ratio in the phenotype of the F2, or he could take the F1, he could do a test cross where here's the F1 being questioned, and the tester would be fully homozygous for the genes and therefore traits of interest, little r, little y, little y. That F1, as we said before, can make these four types of gametes. The tester can only make little r, little y because that's all it has in its genome. You can set this cross up to see what happens using a Punnett square that is 1 by 4. 4 to account for these gametes, 1 to account for this class of gamete. And what you will see if you do that test cross and you go through and assign the genotypes and the phenotypes is that you see all four phenotype categories seen in the F2 a moment ago, but the ratio changes. So in the test cross, you should see a 1 to 1 to 1 to 1 for each possible category of phenotype. So to put a little bit of a human spin on all of this Mendelian genetics conversation, here's an example of a single gene trait that is a Mendelian trait. It is inherited in a way that follows the patterns that Mendel saw back in the 1800s. This trait is oculocutaneous albinism. There are actually many forms of albinism. Oculocutaneous albinism is a Mendelian trait controlled by a single gene. The phenotype of albinism means that you do not produce melanin, and so you have very pale eyes, hair, and skin. You are also susceptible to skin cancer. This condition is associated with having a non-functional tyrosinase enzyme. So you are albino if you don't produce melanin. You don't produce the pigment known as melanin because you don't make working tyrosinase. It's a genetic condition. And again, we're focusing on oculocutaneous albinism type 1. So if you were trying to put genetic symbols to the genotype of an albino individual, you might choose the letter A, and you might say that the big A dominant allele is the wild type gene or allele that produces working tyrosinase. So big A genotypes will not be albino. Little a, fully recessive, will be albino because they have two mutant non-functional tyrosinase genes, no melanin. The carrier heterozygote big A, little a individual has one working copy of the tyrosinase gene, and that is enough to get enough melanin to have a normal non-albino phenotype. But they can transmit the little a allele to their offspring. That tyrosinase gene is located on chromosome 11 in the human genome. Here's another example of a human genetic condition that is Mendelian, and that is cystic fibrosis. There's a gene that encodes a protein called the CFTR protein, cystic fibrosis transmembrane regulator. It's shown in the lower part of this figure here. If you have the normal form of that gene, the wild type form, you have a working CFTR, and it means you manage chloride ions appropriately and therefore have normal mucus in your body. If you have cystic fibrosis, it means you have two mutant copies associated with cystic fibrosis or disease of the CFTR gene. Therefore, you only have mutant CFTR protein. You don't manage chloride ions appropriately in your cells. And as a result, one of the things that you experience is the formation of thick mucus, and that builds up in your your blood vessels and also especially in your lungs and your respiratory system, and you have cystic fibrosis. The gene for the CFTR is associated in a Mendelian pattern, so the healthy form of CFTR gene is dominant. The disease-associated form is recessive. To have cystic fibrosis, you have to be homozygous recessive for that gene, at least usually. There are exceptions to this, but usually that is true. That gene is located on chromosome 7. So you could study the inheritance of cystic fibrosis or the inheritance of albinism, oculocutaneous albinism, and you would be looking for the patterns of a monohybrid, inheritance system. Or you could study the inheritance of the two things simultaneously if you had a family where both genetic conditions were present, and then you would be looking for inheritance patterns like we just looked at in the dihybrid cross system. Here's an example of some problems that you could work for albinism, or you could replace all of this and say that you're working it for cystic fibrosis. You could also develop problems and look at the inheritance of both with the information that we've shared, that I've shared with you in this video. And that's it.